The first part of the gram staining procedure is to produce a smear. This is done by taking a glass slide and labelling it using a Chinograph pencil. Next, we will take our metal loop and sterilise it in the hottest part of the Bunsen flame. We will then take our metal loop and dip it into sterile water, which will pick up a droplet of water. This droplet is then placed onto our glass slide. The metal loop is then re-sterilised in the Bunsen flame and then is cooled by dipping it into the agar. A small sample of bacteria is then taken. The sample is then emulsified in the droplet of water in a front to back motion. The slide is then left to air dry. Once the slide has finished air drying, it needs to be heat fixed. This is done by passing the glass slide down through the Bunsen flame a few times. The smear is now ready to be stained. The stains used in order from right to left are crystal violet, gram iodine, destaining solution, gram safranin and a wash bottle containing distilled water. First, we will add crystal violet to our smear. This will be left on for 45 seconds. After this time, the stain is washed off using distilled water. Next, gram iodine is added to the smear, again for 45 seconds. After this time, it is again washed using distilled water. The destaining solution is now added. This is left on for only 5 seconds before being washed off using distilled water. The final stain to be added is gram safranin. This is left on for 60 seconds. It is then washed off using distilled water. The slide is then blotted dry, making it ready for microscopic observations. Our slide is observed using the 10 times objective. Looking through the microscope, the image should look like this. An oil immersion now needs to be performed to observe the individual bacteria. This is done by adding 1 drop of immersion oil to the centre of the slide and then rotating our oil objective through the oil to the left and to the right to form a meniscus. Fine focusing through our microscope, we are able to observe the bacteria. A purple stain indicates a gram-positive bacteria. A red or a pink stain would indicate a gram-negative bacteria.